Hello and welcome everyone to the fifth episode of the Oz single cell pandemic broadcast. And I'm really delighted that this is a panel discussion, noting the hashtags down in your screen. And it's an exploration of our Oz single cell community with some of the founding members and some of our really dynamic early and mid-career researchers involved in organising this conference and past conferences. My name's Christine Wells, and I'm joining you from the lands of the Wurundjeri people of the Kulin Nation. So, of course, I pay my respects to their elders, past, present and emerging. And I extend that acknowledgement to the elders of the countries from which you are joining us today, and welcome any Aboriginal and Torres Strait Islander peoples who are with us. So Oz Single Cell, this is the fourth year of what has been a really dynamic conference series in Australia, and it's been a truly grassroots movement to bring a very new type of technology to the Australian research community. And I've been wondering for a while now just what motivated the early organisers of Oz Single Cell to stand up and start promoting something that was pretty fringe at the time. So I'm going to start very briefly by introducing our panellists. We have with us today Charlene Naik. Charlene is an immunologist at the Walter and Eliza Hall Institute of Medical Research. G'day, Charlene.
Good morning, everyone.
He's also a bit of a rock star for those of you who watch ABC, the ABC here in Australia. We have Associate Professor Joseph Powell, who is head of the Garvin Wiseman Centre for Cellular Genomics in New South Wales. Hey, Joseph.
Hi, everyone.
And I think anyone who can see Joseph at the moment will realise that he's also a bit of a rock star, but definitely in the bearded department at the moment.
COVID special.
We have Professor Alicia Oshlack with us from the Peter MacCallum Cancer Centre here in Victoria. And Alicia is very well known to many of you as one of our best and brightest bioinformaticians. She's been driving computational biology in the single cell field for more than 8 years now. Hi, Alicia.
Hi everyone.
Good to see you. We're also joined by Professor Alistair Forrest, who is the laboratory head of the Systems Biology and Genomics platform at the Harry Perkins Institute of Medical Research in Western Australia, and who is perhaps most famous for his role as scientific coordinator of the large Phantom 5 consortiums, which is a massive program consisting of more than 250 scientists across 20 countries. So, Al, does that mean that you're very good at wrangling cats?
I'm not volunteering to wrangle that many cats again. G'day everyone. I'm very impressed by the beard as well, Joseph, so good job.
It's also wonderful to welcome Heather Lee, who's from the University of Newcastle. She's an early career researcher who has recently moved back from the UK, where she developed a lot of the technologies for studying epigenetics and DNA methylation at a single cell level. Welcome, Heather.
Thank you. Hi, everyone.
And last but by no means least is the dynamic Fabio Luciano. Fabio is a theoretical biologist, but he works across immunology, systems biology and infectious disease at the University of New South Wales. He's got a broad range of interests, and I know he's going to bring some exciting perspectives to our discussion. G'day, Fabio.
Good morning, everyone.
So I'd like to kick off just by asking each of you, and in particular starting with Alicia, Charlene and Joseph, what is a single cell and why did you get so involved in it so early? Charlene?
Yeah, so I'd never done RNA-seq of any description before getting into single-cell RNA-seq. Little did I know how difficult it was going to be. But when I came back from my postdoc in Amsterdam, it felt like the natural next step. And I'd seen a couple of papers come out. And I knew this was going to be revolutionary, but it really wasn't on anyone's radar that I was talking to. And I literally Googled single-cell RNA-seq, found the top hit, bought the reagents and started. And it wasn't until, Joseph, I think it was an Australian Cytometry Society meeting that we first ran into each other. And we'd been invited to that meeting by Rob Solomon as kind of, what's the single-cell RNA-seq business? And there we first realized that we had similar interests. I think that was right, Joseph, wasn't it?
Yeah, that's my recollection as well, Charlene.
Yeah. Yeah, so...
so Joseph, what... Go ahead, Charlene.
I was just going to say, Joseph came from genomics and I came from flow cytometry. And I think we both realized that, you know, this was one of those clashes of the concepts that was going to emerge. And yeah, I guess we needed a community around that didn't exist at that point.
So Joseph, you're a traditionally trained geneticist. Why single cells?
I thought I was going to go in a very different direction there, ChriSNE.
It could have, it may still.
Yeah, so as you say, ChriSNE, I come from a fairly traditional statistical genetic background where, we've been thinking about linking genetic variation to a phenotype like a disease. And my motivation, I guess, for getting into single-cell sequencing when I started the lab, similar to Charlene, was that it, still at the time, but still this is true, it provides the greatest level of resolution of being able to link genetics through to actual function. Why is a DNA variant ultimately causing, disease or indeed any particular phenotype? And so, the best way from my perspective to solve that is to get to the lowest denominator, the lowest unit of biology and the greatest resolution. And so for me, the single-cell sequencing was a logical way to start with that in that space.
Alicia, is it true that you were dragged into this consortium kicking and screaming?
Well, I think my recollection of this is obviously Joseph and Charlene had been sort of conniving together for a little while, and then they took me out for a beer in Hobart. This is my recollection of this, and they're like, we've got this idea for this, we need to build this community. And I'd already started working a little bit in single cell, and if you know any of my other sort of exploits, you know that I tend to get involved in lots of different conferences. And so I was a bit like, oh, I can't do another one. And then, so I was like, nah, not another thing. But I think it was really, for me, Charlene and Joseph, I don't know, enthusiasm and ideas shining through, and they sort of asked me a few times, and so I'm like, yeah, let's just do it. And so I got involved as well before the first meeting that we had, and I'll just say now that it's actually been a wonderful experience to work with Charlene and Joseph on this over those years. We've had a lot of fun doing it, so I'm glad they persisted in asking me to sort of be involved at that time.
It's been a fantastic conference to participate in, and particularly a very strong sense of community-driven ethos, community involvement, and particularly encouragement and engagement with the early career researchers who are really driving a lot of the innovation in this space. And Heather, I wanted to throw to you and just ask, what have been the surprises for you in the Oz single cell community? You're reasonably newcomer.
Yeah, I was so excited to hear about Oz single cells when I was moving back to Australia. I started here in 2017, and that was the year of the first conference. And I was coming from Cambridge, where we had, we already had a community that was focused on single cell technology and single cell biology. And so I was just so excited that there was already a growing community in Australia. And it was fantastic to see that this community also included lots of different scientific disciplines, from computational biology to cytometry, immunology, cancer, a real mix. And then just to see the creativity and enthusiasm of the scientists involved was really fun.
That's fantastic. Thank you. So I'm really interested from the founders of the Oz single cell concept. What have been the surprises for you along the way? Charlene, Joseph, Alicia?
Why don't you go first, Joseph?
Surprises.
Which question was the surprise?
I don't want to sound glib with saying this, but I think like Alicia, I've been involved in a number of different conferences and communities, but there was a high degree of energy, and certainly the in-person meetings that we've had over the last few years, and that willingness of people to sort of openly contribute and share their experience, their knowledge, their access to technology, or their technical expertise in computational science, and do that without building any sort of barriers or very few barriers to be able to share that information. That was a really, really exciting outcome of building the Oz single cell conference and the community that surrounds that. So that was, I think in hindsight, that's a surprise. It doesn't often happen, and so that's been really one of the most rewarding things that has come out of doing this from my perspective.
Tell me a little bit more about. Go ahead, Alicia.
I was just remembering with our first conference was at the Way High, and the second conference was in Sydney, and it was only something like 8 months after. And I think one of the amazing things about those was the amount of progress that we'd made between the first and the second. It was actually quite astounding. When we put the first one together, it was very grassroots, and it was like, who do we know who's done any single cell, and can they speak about anything? And then 8 months later, we're putting on a next conference, and the progress from between those two was quite astonishing, I felt, and very exciting.
And the demand for the conference too. I remember we were oversubscribed very, very quickly. My most vivid memory of that second conference, Joseph, is that you were actually moving into the Garvin Wiseman at the time. You're still unpacking boxes. And so we're really talking about very, very early beginnings of getting the technology embedded down here in Australia. And have you seen much of a growth in people actually using single cell biology from that kind of Garvin Wiseman perspective?
Yeah, so I think, again, this is something that I was reflecting on about, the early stages of single cells and so on. Is, when we think back at the time, there was relatively few sites or labs that had the ability to generate data, and relatively few labs that were experienced in the, in the computational analysis. And so, the ability to sort of share that knowledge and experience and open access to technology was, was an obvious early win, I guess, of the community, but that is now democratized, huge. Technology access is ubiquitous. Most institutes or labs have access, easy access to generating data. And so that growth and the establishment of of this as a technology platform is now, as far as I sort of see it, is almost cemented. And so we've seen this in terms of the scientific focus of the conference, where, you know, in the first year and a little bit in the second year, it was very technology, as Alicia says. Is anyone doing any single cell work? Let's get them to speak. And now we are really focusing on the biological questions. We're doing it in our, here at the Garn Weissman Center, and obviously most labs are as well. So that's been the evolution. We've gone, I think, from just pure technology and technology access to really the utilization of that for, really deep biological questions.
Al, has that been your experience in WA as well? Because I know that you facilitate a lot of technology uptake there.
Yeah, I mean, it was pretty much in the beginning, do we have access to the technology? Now we've got lots and lots of people coming in. They've got specific biological questions. They've got a better idea about experimental design. We still get some collaborators where they think they want to generate single cell data, but they don't really need it. So you have to have that conversation. But it's been broadly embraced.
And I guess one of the questions I've got for you, Al, is coming from a very large technology-driven consortia like Phantom in Japan, do you feel that Australians are lagging behind in adoption of these new technology platforms?
I honestly don't think we are. I think we've been rapid adopters for this. I mean, single cell was on the horizon for 5 or 6 years before we embarked on this. We could see things. So people were using Fluidigm, for example. There were plate-based methods long before this, but it's being able to do massive numbers of cells easily that's transformed things. So no, I don't think we're behind. I think in terms of the computational side, there's been new methods that have actually come out of Australia. I think in terms of some of the EQTL work coming out of Joseph's lab, that is new approaches, new technology coming out of Australia. In terms of new single cell methods per se, there's some done here, but I think we are playing a little bit of catch up in terms of like combinatorial indexing, for example. But I think we're doing pretty well.
I'm going to throw that same question over to Fabio. What do you think? Are we late adopters?
Good morning, everyone. No, we are. I agree with Al. I remember, just to catch up back what Charlene said, that was a meeting where I crashed the party to the SES in Sydney at the Australian Cytometry Society. And I remember vividly Charlene and Joseph having a coffee. And I crashed on that table and I said, guys, you've been invited here too. And that was 4 years ago. And we were already doing things that a lot of people around the world were not doing it. And I can give you an example. In immunology, the field I work on, there were a lot of people talking about, there is ray cells, we don't know what they are doing, we could do index sorting with flow cytometry. And then people said, well, look, we could put one cell in a plate and we can try to extract mRNA. And those times, no one was actually able to do this in cryopreserved samples. All work was done on cell lines or very easy cells to access. And I think in Australia, we're doing a massive work on that part, right? So we have been actually early adopters, and not only in large cell numbers, but topic, which I stress is being very important, is the identification of rare cell types. Single cell is important not only for big numbers, but also for very small numbers, because that's also was impossible to do before single cell technology came in, right? And we've been covering that space as well in Australia. So I'm very excited to say we've actually been on the front, above the average.
Can I cut in there, Chris?
Please do.
I just want to ask you a question. Is it enough to be early adopters, or can we be pioneers, like true pioneers of trying something that no one's ever dreamed before that's possible?
Can you give me an example?
Well, my own experiences from working in Wolf Reich's lab in Cambridge, and he taught me the power of that sort of visionary thinking and convinced me to try and make a library from bisulfite converted DNA from a single cell, which at the time nobody thought was possible, including myself. So to me, that's the difference between Australian research and European research, or my experience of European research, where in that context you did have the opportunity to do that really blue sky project.
So what do you think is the barrier to the blue sky pioneer versus the early adopter?
Yeah. So thinking about that, I think it's the funding environment. Is that fair? What do others think?
No, completely. I mean, well, at Rickon, you had a blue sky 5-year, 10-year project. You had a vision. You're going to go in this direction. It's going to cost many millions of dollars to get there, but if you do, it's majorly transforming the field, and we don't really have that here.
Charlene, I'm going to throw to you. Do you think that's true?
Yeah, so while I agree with Al and Fabio that we're early adopters, I agree with Heather that we're not early innovators as much as we could be. I think Fabio gave a nice example of that innovation, and you know, we were doing some things in our lab that were kind of really out there. But we found that the culture and the funding structure was just not present to really nurture that. And we ran into a lot of roadblocks, I guess, in terms of people's perception of whether this was worth it or not. And I think we need to change that. I really do. And now there's some really great stuff coming out of lots of labs around Australia in the single cell space, where they've cottoned on to the power of it, and now they're really driving it in pretty risky ways. And I think it's not just science, it's kind of an Australian culture in general. I think we need to push the innovation in this blue sky, but of course money talks, so really we need the structures there to do it.
So, you know,
I mean, the other thing that sort of innovation requires is this mix of disciplines, right?
Right.
And I think we have that, so we can tick that off the culture checklist.
I think that the other piece that's a bit missing from this discussion is a willingness to fail. Because if you're going to be really pioneering, then you've got to accept that a lot of your ideas are not going to work first time or even 10th time. And so you need to have a culture of innovation. I think that you need to be able to balance scientific rigor, which can easily tip over into cynicism, which I think is what you were alluding to, showing, and scientific optimism, and drawing more broadly on the optimism that we have. And particularly, I like the Oz single cell ethos, because it's that optimism that I have been seeing coming through in the community meetings. And while I will agree with you that the opportunity to innovate or pioneer can be difficult to find that niche here, there are no shortage of ideas across the coffee tables and across the beer counters. And I think that the increasing demand for the conference really evidences that. So, you know, I'm going to ask you, and Heather, I'm going to ask you in particular, how do people get started, particularly if they're an early career researcher, they're trying to establish their career here in Australia? How do you get started in a frontier technology as well?
Well, I think the first thing is you've got to be brave, you've got to be courageous, and like you say, you've got to be willing to fail. I'd say, from my own experience, having a great mentor that's sort of been through that process before is really key as well when you're at that early career stage.
So critical mass and mentors who encourage innovation or encourage you to go past the inevitable failure, inevitable blocks. Do you have to have that person at your institution?
I think that is much, definitely. Yeah, preferably next door or in the same department. Yeah.
Alicia, what about from a computational perspective? Where do you get started with this space? Is it just a matter of picking up the right software, or there are other skills that need to come to the table?
Yeah, it was interesting for me when you're talking about innovation and funding. I think that one really nice thing about computational is that it doesn't cost nearly as much. And I think, Australia has capitalised on the computational side of things not because of that, but, given that it's sort of a bit cheaper than trying to develop, say, new technologies or something like that. When it comes to people sort of starting out in computational biology, I think it's sort of what I would do is I look at a lot of the status quo. What are people doing? And then looking at how does that fit with the scientific questions I'm asking. And you really need to, I think the best skill of a computational biologist is to understand what an analysis tool is trying to do, what its purpose does, and whether that really matches with the scientific question you have with your own data. And if that fits perfect, then you can go ahead and follow a tutorial that might be out there, say Seurat tutorial or whatever. But if that doesn't fit, that's when you have to do something innovative with computational sort of methodology, or try and stick things together that haven't been stuck together before in an analysis, or even try and develop your own methodology to do something sort of original, some original science with the data that you're getting.
So, Joseph, you are probably one of the Australian nexuses for people wanting to adopt the technology. And what are your tips for entering the Oz single cell field?
Think carefully about the question you want, and don't assume that the experimental design that's needed to address that is the same as what your colleague has just done. We have in the single cell space, whether it's generating data or the analysis of it, as Alicia's are talking about, there are hundreds, thousands of permutations of an experimental setup, a design, an analysis pipeline, for want of a better word. And so deeply understanding the technology or the tools, computational tools, from a practical perspective, and matching that to your biological hypotheses is from my opinion, one of the most important things to make maximum and best use of the field. And there's a lot of there's a lot of entities in Australia, here in the Gulf Centre, obviously down everywhere really, where there are, very open and friendly centres or individuals that will support and enable that, that pipeline.
So is the need to really deeply understand the technology itself a barrier to adopting it?
I think you need to be open to understanding it. It's a generality, right? You even need to collaborate with someone who really deeply understands it. But then if you're going to do that, then you need to also speak a certain degree of common language. I think a purely transactional relationship where you go, right, well, I need this, I will pay money for it, and not really caring about that interface often leaves a lot of things falling through the cracks. So you do need, and both individuals need to sort of, come to that table and start speaking a common language and understanding what the biological question is and what the technology is able help with. So building that relationships with the people that are able to provide the technology or, you know, have the biological questions, that's something that's definitely one of the most valuable things that people can do, in my opinion.
Fabi, I can see you nodding strongly. Did you want to add anything to that?
I just it's exciting, right? Look, I'm not sure what type of people are watching us, but I believe the conversation that we started in the last 5 minutes about the culture change and the courage, those are the keywords I think are important to highlight to people that maybe are watching us, they want to jump into this. This community has been formed to foster those things, I believe, right? Interdisciplinarity, we are friendly. And the issue I want to make the point, when you asked Heather, you know, do you need to have a big guy that pushes you into the blue skies in your institute? Well, let's not forget, Australia is only 26 million people. There are some of those great guys, great women, but the reality is that we are a little bit isolated. So what do we do to have that happen, even if we don't have that person? And I think those things, the cultural change and the culture and the willingness to do things, to try, to give it a go, and to be very open, because I believe this community is what you need, right? There are people that are truly able and willing to help. And in my experience as a mid-career, this is a rare thing, okay? There is a lot of other society without naming, which is very hard to get this community, right? And this is amazing, right? You can drop on Twitter, on anywhere you want, and you get an answer in half an hour, right? I think that's really the important thing, right?
So I wanted to bring up actually a question from the audience, and I'll come back to you then, Charlene, and that's from Dane, who asks, you know, it's all very well to say to people, be brave, but is it really something that an ECR can be expected to do? How much responsibility is there on us as senior researchers to create the environments and allow intellectual freedom for our early career researchers and trainees to succeed? And what can we do to build that innovative culture?
Yeah, it's a great question, isn't it? And the truth is that when I was in Cambridge, I was in a lab that probably had 10 postdocs, and it was that environment that also allows you to be brave, because you know you have stable employment and you have time to get something to work. So it is definitely linked to the culture as a whole. It's not just up to the one person.
Al, what do you think?
Yeah, I think it comes down to the environment. The group leader needs to have brought in sufficient funds and have sufficient, I guess, slack to allow you to actually give it a try. When I finished my PhD, as you may well know, my supervisor gave me free reins to work up a new protocol for RNA-seq. And I tried many different permutations that did not work, and eventually got something that worked. I've seen similar in Japan at Riken. I've seen similar in Brian Lister's lab over here. So it really comes down to the lab having sufficient funds to do it. The problem is our funding cycles are short, and they're very much milestone driven. So you're given 3 years funding and not enough funding, to be honest, to actually do what you said you're going to do in your grant. So if you're a well-funded lab, then you've got more freedom to let people try stuff new.
I think there's an intellectual piece here too that's really important. And one of the things that I try and do for my own early career researchers is to force them to take the time to think. And it's not just all about the doing, but it's also about reflecting, benchmarking, and just dreaming a little bit as well, right? And that can be quite challenging as an early career researcher, to be benchmarking your ideas because you're inexperienced. So having an environment where you can talk about ideas and critique them constructively, I think, is also a really important aspect of innovation. Charlene, I cut you off earlier. What did you want to add?
That's all right. I guess tying a few of these concepts together, you know, my experience was similar to Heather's in Amsterdam, where I find Dutch scientists think like engineers, and I find Israeli scientists also think like engineers in a way that Aussie scientists don't always, like a few do. And I found that quite uncomfortable at the beginning, but breathtaking in the end. Was at the Netherlands Cancer Institute where I was, everyone who was studying genomics and immunology were thinking about it like engineers, and how do we get a tech to answer these questions that traditional techniques don't? And people would spend years developing that technology, and it was really cultural. And I forgot that when I came back to Australia, that's not necessarily how we do science in Australia en masse. And to have that freedom, that time, and the resources to really delve into it is something I think we could foster more in Australia, and we should. And then second to that is kind of going back to this early question of how do you get started? I think, as we all know, as we all found out, you can very quickly get inundated with all of the different techniques that are out there. I remember in the early days, we created this spreadsheet that we shared between our group. Fabio, you might remember. Joseph, you might remember, in this Excel spreadsheet. And every time a new technique came out, we just added a row. And then we put a column of who should do this in Australia, so we could kind of, you know, distribute the pain to adopt them. And we stopped because we just got overwhelmed of how many different things were out there. Now, of course, you know, 10x came along, and the Rhapsody's coming along, and, you know, there's lots of techniques that are now making a lot of those things redundant. However, just adopting the latest bit of kit that's out there, while incredibly powerful and will drive your research, I think the stuff that, you know, the Heather did with the single cell epigenetic stuff, that can really give you that first insight into a problem that no one else has done before. And I would love to see a way where there is better communication around, well, let's kind of defray some of the developmental efforts, but work together towards doing it. And I think if you can create hubs in individual cities where then that you've got that innovation sense happening, and the researchers who don't have to reinvent the wheel and learn single cell technologies, you know, from scratch, can come in, tap into that resource, work together, as Joseph said, to answer a problem, to deeply understand it in each other's language, and then kind of tap out again. I think we're missing that, and I think we could do that a lot better.
Is that something that Oz Single Cell should be aspiring to do?
I reckon, and look, it's something we've talked about on and off for years and years, but as you can imagine, we're all incredibly busy, and doing this as kind of our 5% of time or 10% of time and resources on the side just means it gets kind of pushed down the list. So yeah, I guess I was hoping in the early days, and still am, that there might be something a bit more coordinated in terms of funding and strategy, where we can really, because that's what Aussies do best, is we're a small country, as Fabio said. We might not have the same resources, but we've got this incredible energy and intelligence and passion and culture to work together. So I think there are ways we can harness that. Unfortunately, it comes down to money, but I think we've got the network there to really capitalize on that.
ChriSNE, can I hijack a second?
Yeah, sure.
I just want to make a point. What ChriSNE said is critical. My experience, because I'm a little bit older than the others too, having worked with other fields such as hepatitis C and HIV, that was a strategy that people have adopted about 15 years ago, to go to the government and say, this is a strategic move. It's a technology investment, and it could be one way to go, is to really highlight the concept of strategy, because there could be funding available for that if we, you know, it would be really beautiful and wonderful to have that type of approach, right? But there is a strategic investment.
I think we do have in Australia a few platforms for funding these kinds of activities, and I'm going to suggest that this is a discussion that we probably continue away from the panel discussion. I'm actually going to bring the conversation back to the practicalities of how do you get involved. And Alicia, you know, you alluded to the idea of scoping computationally the field, finding the gap to develop the next computational method. And I know that one of the things your team has been shepherding for some time is this database of different computational single-cell methods. How big is that now?
Oh, I meant to check, but I think it's over 700 methods for analysing single-cell RNA-seq only. So when we get into the other single-cell sort of technologies, DNA, single-cell epigenetics, multi-omics, that database doesn't capture that. So I'm talking about the scRNA tools database, which you can look up, and that's really been put together by Luke Zapier, and he's still maintaining that. And but it's been astounding. And I guess my, going back to what drew me into single-cell RNA-seq was because I've always worked on transcriptomics, and I remember when RNA-seq sort of started, and it was nothing like single-cell RNA-seq. It was so slow. The computational methods were so slow in comparison. We developed some methods early on without ever seeing any real RNA-seq data of our own, only looking at published data and developing methods. So it is a very sort of crowded field in some ways. And it's exciting because it's moving so fast. And I think we've got to sort of focus on, thinking about what the exciting questions are. And being a computational biologist, it's not about just saying, I'm going to make a better method for doing something that already exists. It's saying, how can we utilize this data in a way that people haven't been able to do before, or that's going to answer a biological question that we've had for a long time, but we can now use this data to sort of look at in a different way. So that's how I go about sort of thinking about what to do, or what sort of things to think about and work on.
And there are a few tools which are kind of go-to tools for a lot of people in the community, and obviously the ChriSNE suite is one of those. We're starting to see a lot of Python-based methods coalescing around ScanPy. I know Luke is now part of that community as well. And so is that enough? Is it okay just to jump on as someone who's got data and wanting to analyse it, jump on and explore it heuristically with something like scQ&A?
Yeah, I think so. If you've got data that fits into that, I think it's a great place to start. And I think that with all these tools, a lot of them are very specialised to different things, which is good if you've got a specialised question. But I think also the field is for sort of what we might consider as sort of a standard analysis. They are sort of converging on a few, so maybe half a dozen or so really tried and tested and well-maintained methods for doing sort of standard analysis. And you can see that from looking at other people's papers and what's really been accepted in the literature, and how it's very important to look at tools that are well maintained so that they're updating all the time and that sort of thing. So I think if you're starting out in an analysis, you want to look for tools that lots of people are using, that are being maintained regularly, and that other people are publishing with. And then, you might have your own specialty things that then you might also try for your particular sort of unique questions.
So I want to turn the discussion now to the future of single-cell technology in Australia specifically, but also where the trends are going globally. And one of the great drivers for me for single-cell biology globally has been the development of these large consortia like the Human Cell Atlas and Phantom, which I guess provide critical mass of people thinking about these problems, the different analysis pieces, the capacity to contribute data, and a leadership team who helps bring it together. So Al, I just wanted to ask you, as someone who's been very involved in these consortia, you know, what are the benefits to getting involved and how do you get involved in these?
So, I mean, with HCA, it's fairly easy just to say, yes, I want to be involved. You go to the website, you sign up. But that's not enough. It gives you, it adds you to the website basically, but you can view all of the content online without registering anyway. To be involved, I think you actually need to attend some of these meetings and physically meet people, which is really difficult now that we have COVID.
Well, our HCA is particularly good at not just doing the webinar, but putting panels on it at our time as well as US and UK time. Yeah. Go on.
I mean, I think that what comes from it is an expanded community. So our single cell is a community within Australia. I've been involved in HCA Asia, and this is sort of started out of Japan. First meeting was in Okinawa, second meeting was in South Korea, third meeting in Singapore. And in 2 weeks' time, there's going to be an online meeting in China. Next year, we're hoping we'll have a physical meeting in Perth in November, and we'll be back-to-back with an Oz single cell meeting. We're hoping that the borders will be down by then. But it's getting together of the group of people who want to work together. The ACA Asia one in particular, there's a lot of really goodwill that people want to work together. How do we leverage funds? And I think that's something that we need to work on more, is how to, as Fabio was raising, how do we work together to bring in larger funds to establish this frontier technologies, ensure our continued innovation and leadership in this space. I'm not sure if I've answered the question.
I think you certainly have. I guess I'm really interested in knowing what's coming down the pipeline in those big efforts, technology-wise. Is it still all about RNA?
Oh, no. I mean, it's everything. I mean, okay, in terms of, and it's not just HCA, in terms of what I think is in the future, cheaper profiling, it has to be. We're paying way too much. Full length, there's lots of development in full length being done. Better detection rates, so detecting more genes. And if you think back to microarrays and RNA-seq, early RNA-seq, they went through this sort of incremental, there was an explosion of new methods. They're all competing against each other. Some are a little bit better than others. And then things got a little bit better, a little bit better, and a little bit better. And eventually, as Lucy was saying, it sort of distills down to a few really good, stable methods that everyone is using. We're nowhere near that yet with single cell. It's massive explosion. And also, I mean, the other areas are like the multi-omics and the spatial. And I mean, I went to the last Oz single cell, and I saw the Visium talk, and we've generated our own Visium data, and it's cool, it's shiny. We're getting something shiny out of this, and it's rapid uptake. So that's sort of what I see is coming in the future. I mean, the other thing is actually understanding the biology. So how are these cells working together in multicellular systems? So yes, we know what cell types are there, but they all work together. They work together at the levels of niches and tissues and organ systems. How do they work together? And that's something we're actually working on.
It's not just about the single cell, it's about the cell ensemble. Is that what you're saying?
Yeah, I mean, we work as systems. We're component systems. So each cell has its own specialised role, and for us to work as a multicellular organism, we have to be able to coordinate those activities. And that's being done through ligand receptor signalling, through cell adhesion, etc. And this is all equally important for development all the way through to immune disorders and cancer. So yeah, anyway, that's my pitch for NATME. I'll let someone else talk now.
So is it all just about the transcriptome?
Oh, of course not. Of course not, Chris. Yeah, so I use methods for DNA methylation, but we also link that with chromatin accessibility and gene expression. But for me, the exciting thing is not just, hey, look at this cool trick I can do in single cells, but actually thinking why we're doing this, what we can learn, and how that's actually going to help someone that might have cancer or any other condition. And I think in one sense, although the funding environment here is so challenging, one advantage of that is it really makes you stop and think, hang on, why am I doing this? And we should embrace that pressure to think hard about the biology, especially when the technology is so expensive.
Absolutely agree. So Fabio, in your field, what's the next big thing? What's the technology you're most excited about seeing coming down the pipe?
I think it doesn't exist. Let me explain. I was reading a review in 2016. I think it was Sarah Teichmann wrote it, and it was trying to tell people why you need to do single cell, right? And there was the metaphor of a soccer, for Australia, AFL, pick a game. Imagine you take a screenshot of people playing AFL, right, or soccer. You see 20 players there. Someone is about to kick. So you could probably guess what he's doing. But the reality is you need a movie. You need a movie, and as Al said, with a lot of cells playing together, right? That was 2016. Now, what I think we need to do is to make a movie of the cells, what they're doing, which cell is using the arm, which cell is using the foot, whether they're being, playing fair or not fair, who is going rogue, who is following, all these things, right? I don't think we have a technology that can do that. But I think that to go back to what Heather said, right? Can we do that? Can Australia do that? Can we come up with a multi-omics 3D or 4D actually, because we need time, that really explain why someone is getting cancer? Right? That's what I think we should try to do. And I know it's hard, I know, but that's what I want to do.
That's a great analogy, Fabio. But really what you want is to use Charlene's analogy, the time travel machine. So you want to be able to look at the picture of the football game and say, that guy's going to score a goal in 3 seconds.
So Charlene, did you want to add into that? Because you're driving a lot of this kind of lineage tracing, right?
Yeah, so Heather's alluding to our work on ChriSNE, which is finding the genes that predict cell behavior. So and the time machine concept is either you can do single cell RNA-seq and you destroy the cell, or you can track that single cell's fate, but you can't do RNA-seq on it, and you can't do both unless you have a time machine. So, we're working, as are many labs around the world, working on methods to try and address that. For me, the holy grail is picking up on what Fabio said, which is how can you, in a live animal, without taking a cell out of the body, track where a cell has been and what it's going to do? It's the movie in essence. And I think we're seeing a hint of it now, and I think you'll see it explode in the next 5 to 10 years, is cell history recorders, where you're going to record what a cell has experienced in its DNA and then read it back later. And then if that can also be embedded in the daughters and granddaughters of that single cell, for example, and find out how they're interacting. It sounds really science fiction, but I think we're seeing the smell of those techniques coming out now. And in 10 to 20 years, I reckon we'll be able to play back that movie. It'll be in small components. It won't be the full HD resolution movie, but I think we'll get little snapshots of it.
So Joseph, I know that you're already doing a lot in multimodal single cell biology. How much of a leap is it to take us through to that next dimension of time and space?
I think it's a significant leap, as Charlene's alluding to. I mean, there's there's hints of methods coming through, and obviously ideas being presented, but probably most importantly, people thinking deeply about how they go from that conceptual idea through to the practicality of achieving it. It's not going to be a quick solution, but like many things in science, I mean, it's going to be so transformative if we're able to do this, that I personally just deeply believe that will happen in, I think in a similar time frame to Charlene, 10, 20 years, and that is a long time frame, right? We're used to thinking about 3-year funding cycles, so that's forever in the future, which means it's challenging, and so it's not an easy leap. But more, on a shorter time frame, there's obviously, other panelists here have discussed, I agree completely with the sort of the technological breakthroughs and next, immediate frontiers. The thing I would add to that is the ability to link a genomic readout with a cell phenotype. And the phenotype of a cell is often very specific to the function of that cell in its biological context. So, the ability to read a transcriptional signature or epigenetic signature and an action potential over a membrane, for example, in the same cell. So there are methods that started to do this, making them high throughput, making them easy and robust to use. That will be fantastic. I think that will be really informative for a lot of biology. And, in my own very immediate field, this is not a technological breakthrough per se, but I really deeply believe that we're going to see a clinical translation. We already are beginning to, of cellular genomic technology, whether it's spatial or single cell based. And I think that's going to be really fantastic for the community to see that impact in the healthcare system in real life, and that's beginning to happen, and that will accelerate, in my opinion.
So can I ask you to elaborate on that? What is missing to actually make that clinical translation piece?
I don't think there's a huge amount that's missing. It's the assembly of different pieces of information and the education of a diverse group, a community to adopt these approaches. I mean, we're already seeing a huge take-up of cellular genomics in clinical trials, whether it's for just, feasibility and discovery, or, or for, just purely because it's kind of an exciting way of generating data and, makes that trial look more attractive. But like any clinical translation, you've got the practical aspect of delivering that, right? You have the biology, you have the tools and the solutions, but you have to put that within a regulatory framework. You need to look at the economic business model of being able to deliver that initially in a private setting or whatever, and then hopefully in a public setting. And so that just takes time for people to work through that regulation and that process. I don't think there's no mountains there. It's just rolling hills that you have to get over.
Heather, is the technology good enough for clinical or diagnostic application?
In terms of epigenetic analysis, there's still a really long way to go. It's very expensive. There's also the issue of delivering to a clinician the right amount of data that they need to take an action for their patient. So with epigenetics, that's harder because we don't have such a direct link between where DNA methylation is important and the control of particular genes, if that makes sense. Whereas with the RNA or with the genome, if you have a mutation in a specific gene, it's pretty obvious that that's where the function is going to be important. But I'm super excited to see that transcriptomics is nearing the clinic.
Absolutely. So Alicia, can you comment on what's needed from the computational workflow to feed these kind of frontier technologies into a clinical setting, even a clinical research setting?
My experience with putting things into clinical, into real clinical sort of production is actually very robust and tried methodologies. And it goes into more of a sort of engineering space rather than a sort of research space in my view. But all that preliminary work needs to be done in the research space and then adopted sort of into an engineering space to really get it into sort of that production level for real clinical application. So I think we're at the point where we're doing it in a sort of research setting, and then that can be sort of engineered out into a more production sort of setting. That's my experience of it. I think that I personally think, and I guess I agree with Joseph on this, that, you know, there are some things that we can do and some diseases that we can tackle with what we have now. And with the methodologies that we have and with the analysis methods that we have now, and it's a matter of just, you know, stepping forward with that in certain areas, I think, and it can transform those areas now. And, you know, cancer is, you know, what I'm focusing on at the moment. I think that's a really good area for that, and there's lots of opportunities in the cancer field, but there's lots of other areas as well where this sort of technology can really transform clinical, you know, clinical decisions and clinical workflows.
Can anyone give me an example of how that's working right now?
I could try.
Go ahead.
I think in immunology we see a lot of that too. The translational part is still not fully clear, but there is a lot of trials that are using already single-cell technology to track, for instance, whether the therapy is working or not. In the context of immunotherapy, probably everybody's familiar with this word now, and even actually in coronavirus research, right? I'm working at the Kirby Institute, where there are now a lot of ideas coming out on using this technology to better understand why people get really sick with coronavirus versus not. I think Joseph is doing something like that too. So the examples are really not fully committed to translational research, but they are ready to develop and to generate knowledge that can inform how to actually treat someone and what to do with that patient, right? And I think that's massive, and I guess that's what Alicia was referring to in cancer as well. Hopefully that makes sense.
It does, thank you. If the future of these technologies is viewing biology from these multiple perspectives, different layers of molecular information, plus the history and the prediction for those cells, how do we do that from a computational perspective? And again, Alicia, I'm going to throw that over to you. Integrating data together and making it more useful than any individual part. That's black magic at the moment, right?
Yeah, I mean, when I think about this, I think about how complicated it is just trying to understand a single cell. Like, it's such a complicated thing just in itself, and then you get, you know, thousands and thousands, you know, tens of thousands, hundreds of thousands of cells together, and then you get all these dimensions of, you know, we've talked a lot about RNA, but, you know, also the epigenetic and the genome. How does the genome control that? And it's a really, really complicated problem, and I guess there's a lot of people getting excited about machine learning methodologies and things like that to try and figure out how we can integrate all those signals. But when I think about it, I think about even one cell is really, really complicated. I think it's really exciting. You know, some of you would know that I started out as a physicist, and this is way more exciting and complicated and interesting, I think. I mean, physics is sort of cool too, but, you know, and I think, you know, I just, you know, I don't have the answers to this, obviously. I don't know that anyone has the answer to that, but I think it's got a lot of elements that are very exciting, and it's exciting that, you know, these computational approaches and the computing resources that we have are increasing. at rapid rates, along with these sort of technology, developments that are going to allow us to be able to try and figure out how to do it. But I don't have the answers to how to do it at this point.
So we've talked a lot about the application of single-cell technology to human health and the possibilities for understanding our own biology, but I'm just wondering, biology is a much bigger area than just us. And so what's happening in Australia in non-human models in single cells? Wow.
Okay. I know that there's people doing protoplast work, for example, so plants. I know that there are people interested in doing, I don't know if they've actually done any experiments yet, but doing it on non-model organisms out in the wild. Quite an expensive way to survey the population, but yeah, that's from my end.
Yeah, one of the early applications in Cambridge when I was there for single-cell genome sequencing was trying to study bacterial species that can't grow in the laboratory, so these real strange chemophile or thermophile organisms that you can't expand enough to get genomic material for. So they are very interested in using single-cell or low-input strategies.
One other area is parasitology, and I know a lot of researchers in Australia and overseas are trying to understand parasite behavior, parasite life cycle, invasiveness stages, etc. At a single-cell level now. So I think both pathogens, commensals, all sorts of non-mammalian or non-animal work is going on, and really exciting for those fields as well.
Outside of Australia, in Japan, I know people working on non-model insects, non-model hibernating animals, all sorts of things. So that it's basically going to come down to money. If they've got money to do it, they can do it, and they have to justify that it's the right technology for the question.
With such a lot of biodiversity in Australia, surely this should be something that we are focusing on.
Can I speak?
Give me the money to do it, I'll do it.
Go ahead, Heather.
Yeah, my husband's actually an environmental consultant, and when we were in Cambridge, he used a PCR test, or outsourced to a company that did a PCR test on pond water for great crested newts. So this is an example of environmental DNA that was there in commercial practice and legislature, because in the UK they have so little biodiversity left that everything is protected fiercely. Whereas here, we don't appreciate the biodiversity that we do have.
I don't think we've even catalogued the biodiversity that we have. There's another really important piece to our human single-cell biology, and that's the fact that we don't explicitly look at equity and diversity in the collections of single-cell data that we're generating. And I understand that this is something that consortia like the Human Cell Atlas are just now starting to implement, but what can we do as a community to not just be more inclusive as a community, but also be very deliberate about making sure that we are truly representing Australia in the biologies we're studying?
Do you want to handle that, Chris, or me?
Yeah, go for it.
Okay, so both Chris and me are involved in the HCA Equity Working Group, and I think for me sort of two fundamental metrics are necessary, that, well, actually maybe three, that we have internationally we have authors from many different countries and ethnicities on these papers. They're not all coming out of two countries, and they're all not coming out of. They're not all of one ethnicity on the authorship list. That the genotypes actually reflect the genotypes within the entire world population. So I've sort of pitched the idea, and maybe Joseph's someone who could actually do this. I can't. Is projecting the human cell atlas onto a genotype space UMAP. So you could actually see how well are we covering different genetic backgrounds. And then the last is actually making sure we have involvement. So within Australia, if we're talking about Indigenous involvement, we should have Indigenous researchers engaged. There needs to be a benefit. And I think for that, it's critical to involve the NCIG. And we've definitely got conversations going on there as well.
Yeah, I mean, diversity and equity is not just about making sure that the collections are diverse, but actually, more importantly, about building capability across our whole community. And for me, it goes beyond just the science. I think it's about having the conversations with the stakeholders in our science, the conversations with the communities who may benefit, or equally important, who may miss out on the next generation of precision medicines if they are not included in the conversation now. And I wonder, this is not something that Oz Single Cell has done in the past, but I'm just wondering if this is an area that Oz Single Cell could develop further. Heather, this is something I know you've thought about in various ways. You've had some innovative ideas on this.
I'm actually not sure what you're referring to there, Chris. Obviously, I think
Your
it's...
cylinder. I think I'm referring to your cylinder.
Oh, okay. Oh, that's not public knowledge yet. Stay tuned for next year, our Single Cells 2021. Yeah, incredibly important to include diversity, but it needs to be driven by the Indigenous community, right?
Yes.
Yeah, I'm not convinced that waiting for the underrepresented populations to catch up with us is the right conversation to have. I think it needs to be more deliberate than that. So we've got an exciting technology. We've got great uptake across the wealthy institutions here in Australia. We've got regional universities. Are we seeing participation of the regional universities in a single cell?
Hi.
Yep.
Well, yes, I suppose so, depending on your definition of regional. I'm in Newcastle, so we have several groups here using single cell sequencing. It obviously depends on the bigger network with, across Australia, so it's not independent at all. I think it could be strengthened.
That's one way our Australia-wide network could actually build capability across the smaller regions, right?
Yeah.
Yeah,
and it probably will be extra challenging given the current political environment.
If I could jump in there, I think the Australian Cytometry Society is already doing something in this space with their roadshow, and were before the lockdown taking skills and knowledge to cities and places around Australia in the context of cytometry, and wanted to bring single cell RNA-seq and other single cell along with that ride. And I think there's a relatively easy opportunity and valuable opportunity to do that, even in a virtual environment, to to maybe learn from those of us in the larger cities and towns, to adapt that knowledge to other places. So I think one thing Oz Single Cell could do is create a mechanism or a forum by which we can impart our knowledge of cell preparation, loading particular machines, analyzing that data. I think we can do something in that space, but we need to identify where those places are and try and create a framework around that. I think there's a really important opportunity there.
We certainly need local champions to help us develop that. And so I guess this is an open invitation for people to get in touch and help us work through those challenges. Look, I think it's time for us to start wrapping up this conversation. And we've covered a broad range of topics around Australian single-cell sciences. I did want to put a pitch in for the future-facing activities. So Al, you've already mentioned what's happening with the human cell Asia. Do you want to just run us through those next series of meetings again, please?
Yeah, just so we're tentatively planning a physical meeting in Perth probably in early November next year, depending on school holidays, etc. And this will be either merged or back-to-back with HCA Asia. And I'm hoping that we get these two communities talking more. A few of the Oz Single Cell cohort have been up to Singapore for these meetings, the HCA Asia meetings, and they're great. Very welcoming community there. And I think if we can work together, it gives us strength when we're trying to negotiate with HCA more broadly. So come on over.
Absolutely. So we're on, this is number 5 of our series of podcasts. We have a further 6 episodes coming. We have 2 episodes focused just on immunology. It's such an exciting topic. We have an episode focused entirely on computational biology. There are some frontier technologies coming forward, and a topic on beyond the transcriptome that Heather is moderating. Is that the topic name now, Heather?
I'm just wondering if I can move to the next slide here in the slide deck. So the topic, oh yes, there we go. So the title we are going with is New Technologies Exploring the Complexities of Genetic Single Cells. So this is really looking a bit deeper, not just at the output of the transcriptome and what that tells us about the cell type, but actually how is DNA regulated in individual cells from epigenetics through splicing and to protein as well. So that's next week. And we've got some great speakers there.
Sounds wonderful. Thank you so much. I also want to announce that we will be holding a poster session, and our final episode of the series will be a poster session. So we will be asking all of you to send in your short videos or poster presentations, and we will be announcing on the Oz Single Cell website and through social media how you can participate in that. There will be some career discussions with some of our industry partners interposed throughout that poster platform as well, so it should be a lot of fun. And we also have, of course, a competition for the interpretive clustering competition. I'm going to let someone else introduce that because I'm just not quite sure.
I'll be happy to do that. I'll be happy to do that. So look, for those of you who've come to Oz Single Cell meetings, you'll realize that, you know, the science is great and that's why we're here, but we also want to make this a pretty casual, fun group of people. So a number of years ago, enlightened by Christine's voluntary interpretive dance of a DiSNE plot, we created the ChriSNE Award. And we will be holding it again this year virtually. And we ask people to send in their photos or short videos of their interpretive ChriSNE or CrewMap or whatever the latest dimensionality reduction method is out there. And we're going to play a short video of past entries to inspire you. So you can submit them online, and there will be prizes. And you've got a few weeks to come up with them.
So that's it for our single cell pandemic panel discussion. I'd really like to thank all of the panelists for joining us today. It's been a fun conversation from my end. And thanks to all of the Oz single cell members of the audience who asked questions along the way. I hope you heard your question being pushed forward to the panel throughout the discussion. But of course, if you want to follow the conversation, then please do so on Twitter. The hashtags OzSingleCell and Squander, scQ&A, are on your screen. And a very special thank you, of course, to our sponsors of this episode. We have been really fortunate for OzSingleCell to have great sponsorship across the series. We're particularly thankful for that. So that's it from me. That's it from the panel. We have a short feedback form for you to fill out now, and we'll see you next week for the Beyond the Transcriptome episode. Bye all.
Bye-bye.